Okay, thank you very much for a nice introduction, Kubo Sensei. And first, I'd like to thank Sophion Bioscience and the organizer committees for inviting me to this wonderful symposium. So here I'm going to talk about the structural insight into the closure of a large pore channel in phospholipids by cryo-EM. So first, I'd like to talk about a brief introduction about gap junction channel. So gap junction channels cell-cell communication channel to transport small molecules such as nucleotides, second messengers, ions, amino acids, up to peptides. And this function is associated with various kinds of biological events. For example, heart development, fertility, electrical synapse, immune system, and others. And the loss of function of this gap junction channel is known to cause human diseases like hearing loss, cataract, skin disorders, and heart diseases. And this figure shows a model of a vertebrate gap junction channel. So in the case of the vertebrate, a complete channel contains a connexin dodecamer, 12 subunits. And there is connexin hexamer in one membrane. This is termed hemichannel because of half size of the channel. And there is another hemichannel in the adjacent membrane, and two hemichannels are docked face to face to be a complete gap junction channel. And this is often termed large pore channel because the pore size is larger than 10 angstrom. It's kind of big. And connexin contains 4 transmembrane helices. And I've been interested in the gating mechanism of this channel. That is how to close this large size of the pore. And a long time ago, we presented a low-resolution 3D reconstruction of connexin 26 mutant structure using electron crystallography. And here we found a physical blockage in the pore. And based on this, we proposed a plug gating mechanism made by N-terminal portion. And later, Osaka University group reported X-ray structure of connexin 26 wild type. And this structure shows the N-terminal arrangement in the pore forming the funnel conformation. And so based on this, we interpreted this probably represent open form. And today's my first topic is not connexin, but inexin. Inexin is invertebrate gap junction protein, which has the same membrane topology as connexin. However, there is no sequence similarity to vertebrate connexins. So this shows the inexin family tree. And here I focus on C. elegans inexin 6 protein because only this isoform successfully made oligomeric channel in the insect expression system we used. So several years ago, we presented the atomic resolution structure of inexin 6 gap junction channel in detergent using a single particle cryo-EM. And this was the first atomic model in the inexin family, and the map quality was sufficient for de novo atomic modeling. And in structure, we found the C. elegans inexin 6 and human connexin 26 X structure show a very similar structure arrangement in terms of monomeric and oligomeric form. However, the subunit number is different. Inexin contains 16 subunits, while connexin 26 has only 12. And this structure shows a very wide pore pathway, of which the diameter is about 18 angstrom. So this is surrounded by N-terminal funnel conformation and looking like an open form. So this N-terminal funnel conformation is the same as one which was found in a connexin 26 X-ray structure, and also found in recently reported a cryo-EM structure of connexin 46 and 50. So we think the N-terminal funnel is probably conserved in the gap junction family protein, and it's essential in the channel opening activity. And the remaining questions are, how can this wide pore be closed, and what is the structure in lipid bilayer, and is there any difference between duct and hemichannels? And as a next step, we investigated structures of unducted inexin 6 hemichannels in phospholipids. For this purpose, we used the nanodisk reconstitution technique. First, inexin 6 hemichannels are purified in detergent and mixed with lipid POPC and MSP2N2 belt protein And this mixture was incubated with biobeads to exclude detergent. And this resulted in the formation of nanodisk reconstitution. And nanodisk particles were confirmed by negatively stained electron micrographs, and we employed cryo-EM for high-resolution structural determination. The final 3D reconstruction was determined at 3.8 angstrom resolution. And the overall structure of inextrinsic hemichannel in nanodisk in magenta is consistent with the duct junction form of inextrin in detergent colored in green that I presented in previous slides. So the extra region of the structure in nanodisk are partially disordered. This is probably because there is no interaction with opposed hemichannel as a docking counterpart. When looking at the sectioned view of the cryo-EM structure, we found some distinct features. First, there are new densities termed X and Y inside and outside the pore, which are located at the near pore entrance. The other one is there are double layered densities in the pore obstructing the pathway. So this double layered density is at the level of the transmembrane region. So we assumed this could be the partially resolved head group of the phospholipids. However, all these newly observed densities are too ambiguous to assign the atomic model. So we obtained another two structures of the cryo-EM by cryo-EM. The first one is the N-terminal deletion, where amino acid residues from number 2 to 19 are taken away in nanodisk. So this deleted peptide corresponds to most part of the N-terminal portion of the Y type, which are associated with forming a funnel. So please note that this N-terminal deletion can no longer form N-terminal funnel in the pore. The another structure is Y type, but still in detergent. So this transmembrane region is surrounded by detergent micelle. So first, when looking at the structure in detergent, a Y-type structure in detergent, we found the N-terminal funnel configuration looking like an open form. This is consistent with the duct junction form in detergent. So given this, newly observed densities found in nanodisk probably contributed by the nanodisk reconstitution. And also, when looking at the N-terminal deletion structure in nanodisk, we found double layer of pore obstructing density a little bit ambiguous though due to a lower resolution. However, the densities for X and Y found in the Y-type nanodisk structure did not appear in N-terminal deletion. So given this, density X and Y probably contributed by N-terminal portion of inextrin. And eventually, these double layer densities are produced by the nanodisk reconstitution rather than N-terminal portion. And we also obtained another two cryo-EM structures of duct junction form in nanodisk of Y-type and N-terminal deletion. And both structures show the double layer pore densities in each pore cavity. Specifically, when looking at the structure of N-terminal deletion, we found some extending density from one of the layers. So this could be a partially resolved acid chain and supporting the idea that phospholipids may exist in the pore. And so when this inextrinsic hemichannel is in detergent, we found a very wide pore pathway surrounded by N-terminal funnel suggesting an open form. And when this channel is in nanodisk, we found N-terminal rearrangement along with pore obstructing densities in double layer. And at present, we assume this could be the phospholipids located in the pore. However, we do not have good way to label the lipid molecules in the cryo-EM structure. So further structure study will be required to make sure this interpretation. My next topic is about pannexin channel. So pannexins are vertebrate proteins categorized as gap junction family, but it has only a partial sequence similarity with the inexin, and there is no similarity with vertebrate connexin. So we can say that pennexin is a mammalian innexin homolog protein. The topology of the pennexin is the same as the other two members, containing 4 transmembrane helices, but these proteins, pennexins, are not thought to form junction because there is a glycosylation site in the second extracellular loop, and this is going to be a hindrance for junction formation. And for human, pennexin, 3 members, pennexin 1, 2, 3, have been identified, and pennexin 1 has been the most studied and well characterized. So it's been reported that pennexin 1 channels release ATP, and this results in the inflammation or apoptosis and cancer cell proliferation and epilepsy. And it is known that the C-terminal truncation of pennexin 1 constitutively activate the channel. And so this C-terminal cleavage is mediated by caspase-3 in vivo. And recently, many groups have reported pennexin 1 cryo-EM structures. And all structures determine the one single channel contains 7 subunits, and mostly determined in the detergent except for 2 cases. One used nanodisk, and the other one used a lipid disk. However, no study has shown the conformational change between open and closed form in lipid bilayers. So we studied the structure of the pennexin 1 in lipid nanodisk, focusing on different configurations. So here we expressed human pennexin 1 proteins and EXP293 mammalian cells. And here we expressed wild type and N-terminal deletion and a C-terminal deletion. So this C-terminal truncation site is consistent with the caspase-3 cleavage site. So after removing the C-terminal tag, pennexin 1 purified in the detergent were reconstituted into nanodisk, as done with innexin 6 hemichannels. So negative stain electron micrograph confirms a nanodisk formation, and we performed the cryo-EM. So here we determined the 4 pennexin 1 structures in total. First one is wild type apple, and the second one is wild type, but in a buffer containing a blocker. This is termed the probenecid. And we call this conventional pennexin 1 occluded. And the third one is C-terminal deletion, and finally N-terminal deletion structure. And this slide shows the structure of wild type apple form, confirming the 7 subunits as reported previously. And this structure shows a wide pore surrounded by N-terminal funnel conformation. So looking like open form. However, the pore constriction is located at the extracellular region surrounded by extracellular loops. And diameter here is about 7 angstrom. And interestingly, there are unassigned density on the pathway. So we believe this N-terminal funnel probably represents the open form. However, I have to say it's unlikely, it's finally unclear if this structure represents a physiologically open form or not. In this structure, we observed the lipid acyl chain densities in the subunit interfaces in the transmembrane region. So these lipids have the interaction with the transmembrane helices through hydrophobic residues. And N-terminal portion containing short helix forming the funnel, separating the pore region and the hydrophobic transmembrane region. This N-terminal helix also has interaction with TM1 and transmembrane helix 2 through hydrophobic residues. And this slide shows the pennexin 1 wild type, but in the buffer containing a blocker probenecid. So in this reconstitution, N-terminal portion is oriented to the cytoplasmic side, so taking a different conformation. However, finally, we could not determine the density corresponding to a probenecid blocker. So I have to say, if this conformation, it's unclear if this conformation is directly fixed by a blocker or by other factors. And in this reconstruction, we also found a double layer pore density. This is consistent, similar to the pore density found in the indexing 6 hemichannels in nanodisk. So that's why we call this the pannexin occluded. And also the density for lipids found in the intercellular species. But not only here, we also found the lipid acid chain densities inside the pore, along with transmembrane helix 1. So there are lipid densities inside the pore and outside the pore and the intercellular species. So we assume the lipid can penetrate through the intercellular spaces at the lateral movement. And this slide shows the N-terminal deletion and C-terminal deletion structure. For N-terminal deletion, there is a pore occluding density a little bit ambiguous due to a lower resolution of 4.5 angstrom. But this property is consistent with the pannexin 1 occluded form. And for C-terminal deletion, we found N-terminal funnel conformation looking like open form and consistent with the wild-type apple form. So this is a functional assay using the Xenopus oxide voltage exam method in collaboration with Dr. Watanabe in Osaka University. So for wild-type, colored in red, and the C-terminal deletion in magenta, produced a positive current depending on the applied membrane potential. And these positive activities were suppressed by a blocker probenecid. So this channel opening is consistent with the cryo-EM structures of these two constructs, showing N-terminal funnel looking like open form. On the other hand, N-terminal deletion did not show any positive activity. And this revealed the dominant negative effect against the wild-type co-expression, and also a double mutant containing N-terminal deletion and C-terminal deletion also did not show any current. So this suggests the intact N-terminal portion is required for a channel opening of the pannexin 1. And here we also employ the MD simulations of the pannexin 1 channel embedded in the POPC lipids. This calculation is performed by Dr. Terada in the University of Tokyo. So after one microsecond calculation, the lipids in the intercellular spaces in upper form, for example, the yellow one migrated toward pore. However, this movement is blocked by the N-terminal funnel conformation. And also the green one found in occluded form leaving from the intercellular space. Instead, the red one came into this space. So this suggests the lipids around the pannexin 1 peptides are flexible. However, the lateral movement of the lipid passing through the intercellular space was not observed during this calculation. So the interpretation of the lateral diffusion still remains to be elucidated. So in this study, we found the two conformations of the pannexin 1 channels in a lipid nanodisk. One is wild-type apple, showing the wide pore surrounded by an N-terminal funnel. And the other one is pannexin 1 occluded form. And here, N-terminal portion is oriented to the site-specific side, and there is a pore occluding densities. And at present, our interpretation is there are phospholipids in the pore and may be able to migrate as a lateral movement. However, this interpretation still, I have to say, it's still speculative. And interestingly, other groups have reported similar interpretation structures. For example, LLC8NC, CAM channel, and connecting 36. cryo-EM structures shows pore density, and some of them show a double layer, and lipid gating mechanism has been proposed. Totally the same as our interpretation. So hopefully, it would be nice to see more rigid evidence to show these pore densities are not artificial, but physiologically significant. So here we obtained the cryo-EM structures of indexing-6 hemichannels and peniximon structures in nanodiscs. So we think protein-lipid interaction would be more and more important for understanding the gating regulation of gap junction family protein and large pore channels. So thank you very much for your attention.
Thank you very much for beautiful presentation. And any question, comment? Yes? Please say your name and the institution first.
Yasushi Okamura from Osaka University. Thank you very much for wonderful talk. I will pick up later. Thank you very much for wonderful talk. My question is, your lipids found in the structure came from the native tissue in the heterologous expression system, or when you made a nanodisk, I think you added POPC. So was a POPC added during nanodisk formation shown in the structure? That's the first question.
Yes, I can't say the conclusive interpretation, but the possibility is carryover lipid may exist in the intersegment species, because it's been reported that the solubilized form, not in nanodisk structure, also reveals the similar lipid densities in the intersegment species. And but in the solubilized form, I think that no density was found inside the pore. So in the case of the pore occluding density, I think it's more likely in the added POPC.
Likely added POPC.
Likely, yeah.
Okay, thank you very much.
Yeah.
Any questions? Yes, Alex, please. Jianmin first, and then Alex.
Oh, okay. Yeah, so that's interesting. I'm not knowledgeable enough. Can you tell me that with the lipid closed conformation and the open conformation, is there any transition between those two conformation in the physiology?
I don't have a clear answer for your question. And what I can say is the observed structure is something like this, and the problem is if it's possible to take this kind of conventional change without any using, for example, the ATP hydrolysis energy, because this is channel, there's no such a positive driving force. So yes, at present it's not conclusive, but yes, observed cryo structure reveal this property. So I should say it's possibly artificial could be included. Yeah.
Thank you.
Sasha Sobolevsky, Columbia University. Very nice talk. I'm here.
Yeah.
I'm not gonna stand up because something will fall off.
That's okay. Yeah.
So you showed very nice molecular dynamic simulations. And you showed us behavior of the lipid. Have you looked at the behavior of ions and water? Because you have two different sizes of the pore at minimum, right? And do you see difference in water and ion conductance through the channel?
During that calculation, you mean that the
during the molecular dynamics?
Yeah. And between the upper and the occluded form?
Right.
We don't see the clear difference between them.
Right.
Yes.
Thank you.
Okay. Then one last question.
Thank you. Anna Moroni from University of Milan. So I was wondering, what is the conformational change that makes the pore hydrophilic or hydrophobic? So do you see movements that turn the side chains or something? I'm
not sure if there is a clear property about the pannexin, but it's basically N-terminal helix, as I said, forming the funnel. So the surface of the pore would be hydrophilic, and the orientation facing to the pore wall would be hydrophobic. So when forming the funnel, it forms a hydrophilic pore, and if N-terminal is oriented to the cytoplasmic or taking conformational change, it's going to be the hydrophobic surface will be appear.
Okay, it's almost time. Thank you for your beautiful presentation.
Thank you
again.
very much.